Okay, ladies, so with this little video, we're going to try to review the various parts of the eukaryotic cell that we introduced last class. And through this video and an activity that we will be doing next class, we are hopefully going to review these structures well and get them back in your heads. I know that all of you at some point have learned most of these structures. Usually most of you will have probably made a model of a cell back in middle school or something, and so hopefully this is all sounding or will sound kind of familiar to you. Now we're going to start with the nucleus, and with all these structures we are going to look at the cell as though it is a city. So it's going to be our analogy that we're going to use, is the cell is a city. And so each structure, as we learn its function, we're going to compare that to what the function would be in a city. So the nucleus is our control center, right, of the eukaryotic cell. It is a centrally located structure surrounded by a double membrane, and we call that the nuclear envelope. There's a bunch of pores in that envelope, actually, so it's got holes through which material can enter the nucleus or leave the nucleus. Inside of a normal nucleus, you see 3 nuclei down here. So we've got 3 different cells. Kind of outline a cell here, and then inside there is going to be the nucleus in that darker red. And you notice that inside here it's kind of splotchy. That grainy or splotchy material is called chromatin, and this is a mix of DNA and protein that we find in the nucleus. Now when a cell begins to divide, which will be the topic of our next unit, the chromatin will actually pack together more tightly to form chromosomes. And you might remember learning about these in the past, but they sort of have like an X shape to them usually, and that only happens when the cell prepares to divide. So if the nucleus is a control center, oftentimes what we can say is that this is sort of the mayor, or even maybe just the mayor's office in a city, right? Because it's in charge of telling the cell what to do and keeping everything in order. And that's basically what the nucleus does. The nucleus contains that set of blueprints, the chromatin, that tells the cell how to do its job. Now our second structure is actually located within the nucleus, and it's called the nucleolus. So notice the spelling here. This is not a nucleus, right? This is nucleolus. And this is a smaller, dense region inside the nucleus where we see a high concentration of RNA, which is in one of the other forms of genetic material. Now the primary function of the nucleolus is to build ribosomes. So this is where we make ribosomes, which is another structure that we'll learn here in just a moment. So if you notice in this picture, our cell would be out here, right? And then here's our nucleus. Notice all the pores and everything. And then inside that nucleus, you've got what's called the nucleolus. Now don't let this throw you off. A lot of times people like to say, well, if the nucleus of the cell is like the mayor's office, then the nucleolus is the mayor or something like that. That's totally not correct, right? The nucleolus is simply there to build ribosomes. So whatever the ribosomes are in our analogy, the nucleolus would be the thing that creates those. So our third structure here is the ribosome. And the ribosome is really important. Ribosomes are found in really every cell in nature, and they are a solid cell structure, so they are not a sac or a bag, kind of like a nucleus, but they are solid, and they make proteins, and this is super important. So if you learn anything from this video, make sure you know that ribosomes make proteins. Here's a picture of some ribosomes. The way that ribosomes work is they're this solid piece of RNA and protein, and they actually can read genetic material, in this case RNA, and use that information to build a protein. So what we see here, it looks kind of like beads on a string, And the string here would be that RNA, and these ribosomes are actually sitting on it and making proteins from it. Ribosomes are like machines in a factory. They take directions from the nucleus, actually, and then they build that important product. Ribosomes sometimes are just loose in the cell, which is what we see in this picture, or they might be attached to another organelle called the endoplasmic reticulum, and so that's where we'll go next. So ribosomes in our city are some kind of factory or machine that makes a product. So whatever our product is, let's say in our city that we are producing One Direction t-shirts, okay? And it can be any direction you want, north, south, east, or west. Get it? Isn't that joke great? Now, so we're going to make a One Direction t-shirt. And so that's our protein, is the t-shirt. Right, that equals protein, which means that then the ribosome is the machine that makes this. So if we have a machine that makes these t-shirts, that's our ribosome. Now we said that ribosomes can be attached to the endoplasmic reticulum, or sometimes much more simply called the ER. The ER is a set of membranes which kind of form these tubes or sacs around the nucleus. So notice in this picture, here's our nucleus, and then the endoplasmic reticulum kind of fans out from the nucleus in these little sets of membranous sacs or tubes. Now, the ER is where proteins and fats or lipids get packaged, altered, and prepared for transport in the cell. So let's say that we do make our little One Direction t-shirts. Okay, here's my British guy, and we make these in the ribosome, right? And then from there, we start out, maybe let's say we just make a plain t-shirt. So I should not have drawn the British guy in the middle of it. We make a plain t-shirt, and then we're going to pass that on to the ER, where it is packaged, altered, and prepared. Let's say, so when we go to the ER, we maybe put on the little decal of the British person. We add maybe some other details to it, and then we also put a price tag on it, right? And we ship that out to our to the next step. So that's sort of what the ER will do. The ribosomes make the proteins, or other things make lipids, and then they get processed here in the endoplasmic reticulum. So ribosome makes our product, sends it to the ER where it's going to get further processed and then move forward. There's actually two types of endoplasmic reticulum, as you might have noticed on the last picture. Some of this we call rough ER, and the other part we call smooth ER. Now the rough ER is involved in synthesizing and modifying proteins. It looks rough, like we see in this picture and in this real picture here, because it has ribosomes stuck to it, or it is studded with ribosomes. So the rough ER deals with proteins. The smooth ER looks smooth because it does not have ribosomes stuck to it. That's the main difference. And without ribosomes stuck to it, that means this part does not process proteins, but it tends to process lipids and help you make fats. Now, this is where structure and function can really start to tie together. What we know, based on these things, is that the rough ER, which makes proteins, is going to be in cells that make a lot of proteins. This would include digestive cells that would release your digestive enzymes. This would include the pancreas, which releases a lot of proteins, or things like that. The smooth ER is going to be found places where we make a lot of lipids. Now, one of the primary reasons that we make lipids in the human body is as hormones. So hormones like estrogen or testosterone those are actually lipids. And so let's say, the ovaries, which is the organ that creates those female hormones like estrogen, the ovaries are going to have a lot of smooth ER because they have to make a lot of those lipid hormones. So both of these structures are found in most cells, but we might see more or less of one type or the other, depending on what the cell does. The next step is the Golgi apparatus. So if we made our product in the ribosome, then we modified it some in the ER and processed it, then we're going to pass it on to the Golgi apparatus. So this is another set of membranes that form sacs. This comes after the endoplasmic reticulum. A lot of times we say it looks like a stack of pita breads or something like that. Proteins that form in ribosomes get processed in the rough ER, then they move to the Golgi, which further modifies, sorts, and packages the materials. So the Golgi's job is to prepare the materials so that they can be transported around the cell or secreted, which means released, out of the cell. So this is the shipping center that makes sure packages get shipped correctly. So the Golgi is like UPS, right? We're going to make our t-shirt at the ribosome. The ER is going to get the price tag on the t-shirt. It's going to get the decals or whatever on the t-shirt. Then we're going to send it to the Golgi, which is going to pack those t-shirts into boxes and send those boxes wherever they're supposed to go, right? Out to the stores or whatever it might be. And we rely on UPS to make that shipping happen. And so that is sort of the Golgi apparatus's job, is to further modify and package everything and get it to where it's supposed to go. Now, we move away sort of from this line of protein packaging things to some other organelles. The first one being a lysosome. Now, a lysosome is basically a bag of chemicals. Okay, these are enzymes which can break things down or cause chemical reactions, like we learned back with the chemistry type stuff. And these enzymes, in particular in the lysosomes, break down or digest materials. Now, don't think of this like food digestion in your stomach. This is digestion at the teensy tiny cellular level. But these lysosomes can break stuff down within the cell. This stuff might be lipids, it might be carbs, it might be proteins, it might be organelles, it might be other parts of the cell. If this is an immune cell, it might be breaking down bacteria that it takes in. So lysosomes can really basically destruct anything that they want to. And so we oftentimes think of these as the cleanup crew of the cell. This is the garbage men or garbage people in our city that make sure that everything gets cleaned up when it needs to and broken down and gotten rid of. Another organelle is called a vacuole. Vacuoles are big bags that we use for storage. So vacuoles can store food particles, they can store enzymes, they can store any sort of chemical. They very often might store water in the cell, particularly in plants. So plants have a special vacuole called a central vacuole. And in a plant cell, a lot of times that central vacuole is just huge. It actually takes up most of the cell. And that's usually there to store water. Animal cells also have smaller vacuoles, though, which is what you see here. And you see these little shapes here inside the cell. Those are all vacuoles in an animal cell, so much smaller. Vacuoles, simply put, in a city are anywhere where you can have storage. So maybe they're like those self-storage places that you see on the side of the road all the time where you can store your stuff. Now, another super important organelle is called the mitochondrion. Mitochondria is actually plural. One of these is called a mitochondrion. And we're going to find these in all eukaryotic cells. And they are the places where we can convert chemical energy from foods into energy that the cell can use. So, in other words, they are sort of the energy factories of the cell. Now, they are enclosed by a double membrane, as you see in this picture. There's an outer membrane that's thicker, that's there for protection, and then there's an inner membrane which is all folded up like you can see here in this one, and takes part in those energy reactions. And we'll learn about those later on after Christmas. But it has sort of a strange structure. Now the big thing to remember is energy, right? This is the energy producer. So if we're talking about this in terms of a city, we would say this is the LG&E. This is the power plant that this cell uses. Now it's much more complicated than that at the cellular level, but that's enough for now, and we'll learn that more complicated stuff later this year. Very similar to a mitochondrion, but only found in plant cells, is the chloroplast. Now, chloroplast we often say is found only in plant cells, but that's not quite true. Chloroplast can be found in some types of fungus, some bacteria. The important thing to remember is that chloroplasts are never in animal cells, right? You're never going to see a chloroplast in an animal cell. And a chloroplast is the organelle that captures energy from sunlight and turns it into chemical energy for the cell via the process of photosynthesis. So chloroplast is where photosynthesis happens. These are green structures because they contain a chemical called chlorophyll, which is green in color. They also have a double membrane, sort of like the mitochondrion on the last slide. And this organelle performs, I'm sorry, it's kind of cut off, autotrophy, right? So this is what makes you a producer, is if you have chloroplasts. Not really sure what you'd say this was in a city. Maybe a chloroplast is like a solar power plant, I guess, that uses solar panels and sunlight to make energy. The big thing is that they can do photosynthesis, and we'll learn more about this structure later this year too. A couple interesting things about mitochondria and chloroplasts before we move on. Mitochondria are totally inherited maternally, which means that all mitochondria come from your mom and not your dad. This will be more significant later this year when we talk about genetics, but it's just kind of cool. So you can go find your mom and thank her for giving you all of your energy because she's the one that gave you mitochondria and not your dad. The other thing that's kind of interesting is that mitochondria and chloroplasts both have their own genetic material. So while usually the DNA is inside the nucleus, we actually see a little bit of DNA found within this organelle. What this shows us, and we'll talk about this later, is that these cells were probably, these organelles, this mitochondria and chloroplast, were probably their own cell at one point, and somehow got incorporated into a larger cell and became an organelle instead. Now moving on, we've got the cytoplasm and cytoskeleton. Cytoplasm is the part of the cell that is inside the cell but outside the nucleus. So you sort of have this jelly-like space inside the cell. Here's a cell with a nucleus. You got this sort of jelly-like stuff, and that's part of the cytoplasm. But pretty much anything else in the cell could also be called cytoplasm. So anything in this area out here that is inside the cell but not the nucleus is called the cytoplasm. The cytoskeleton is a set of proteins that sort of forms a scaffolding or a framework for the cytoplasm. So this cytoskeleton, just like our skeleton is the frame our body is shaped around, the cytoskeleton is a framework that the cell is built upon, and it allows movement of the cell, allows structure of the cell, keeps the organelles where they're supposed to be. The cytoskeleton is made up of little proteins called microfilaments and slightly larger proteins called microtubules. Another structure is called the centrioles, and they are pictured here and here. Use a different color here, circled. The centrioles are little organelles that help to organize the microtubule proteins. So that's what these dotted lines are meant to be, are microtubules. Now this picture might look a little bit familiar to you, hopefully, because it's showing you part of mitosis, or cell division, that we'll learn about a couple weeks from now. The important thing for now is to know that these centrioles help frame up the microtubules to form what's called a spindle, or mitotic spindle, which is very important for cellular division, which this is no longer true. We're going to learn about this right before winter break, so just a couple weeks from now. Now, one of the last things that we want to look at are the outside structures of the cell. Every cell, no matter if it's a eukaryote or a prokaryote, has to have an external barrier, right, that defines the edge of the cell, and that is called the cell membrane, or sometimes called the plasma membrane. The cell membrane is there to protect and support the cell, and it also helps determine what can and cannot enter or leave the cell. So we call the cell membrane a selectively permeable structure. Permeable meaning you can get through it, but selectively meaning only some things can get through it, right? So it lets some things through, but not others. What this tells us is that the cell membrane for our city is sort of more than just a big fence or a big wall around the city. It actually is selective in what it lets in or what it lets out. So I don't want to be political at all, but in terms of the Border Patrol, which helps keep people from coming into our country that shouldn't be there, that's sort of what your cell membrane is like. Not only is it a wall or a barrier, but it also makes decisions on what sort of molecules it lets into your cell and what kind of molecules it lets out of your cell. A membrane is mostly lipids, and the number one kind of lipid is called a phospholipid, but it also has proteins in it and carbohydrates in it as well. Now, as I said a second ago, the major component of the cell membrane is called a phospholipid. So this is a special type of fat that has this head group, which has phosphate in it, which is where phospho comes from, and then these two fatty acid tails or legs that hang down. Now, phospholipids are unique. This is very important right here, because they have heads that are hydrophilic, but tails that are hydrophobic. And I think you guys might recognize those word roots, and we've already kind of talked about this at one other point earlier this year. Hydrophilic means that they love water. So the head region of one of these phospholipids really likes water, or is attracted to water. Whereas the hydrophobic, or water-fearing, tails do not like water. So they don't like water at all. This is important because it allows the membrane to set up in this way, where you form a lipid bilayer, or two layers, where the hydrophilic heads are directed toward water outside the cell. Hydrophilic heads also directed toward water inside the cell, and these hydrophobic tails can hide in between. So the hydrophilic heads go toward water on either side, and the hydrophobic tails can hide in between. Now, there are two types of proteins that are also seen floating around in this sea of phospholipids, as we usually kind of draw it. And these proteins are named based on where we find them. If they're only attached to one side of the membrane, we call those peripheral membrane proteins, meaning peripheral meaning along the edges, right? So if you go to a volleyball game, right, everybody's sitting at the periphery of the court. They're around the outside. So peripheral membrane proteins are only on one side of the membrane or the other. Other proteins actually extend through the membrane, stretching from one side to the other, like you can see these guys right here, and we call those integral membrane proteins, right? They are within the membrane, so we call those integral. Now, a lot of these are just like this one right here, which forms a channel through which stuff can pass through the membrane. So, in other words, it's kind of like opening a window or a tunnel through the membrane for molecules to go through, and we'll learn more about that next week. Those carbohydrates that we mentioned that are also part of the membrane are usually attached to the outside. They help identify the cell. So you have different kinds of cells, right? Blood cells, brain cells, muscle cells, skin cells. All of these are going to have different kinds of carbohydrates on the outside, which is going to identify the cell as whatever type it is. So a blood cell is going to have different carbohydrates than, say, a skin cell would have. Last little thing about cell membranes is that scientists often say that cell membranes follow the fluid mosaic model. This means that the membrane is fluid. So again, this is why we kind of often see it drawn like this wavy ocean of lipids. And that means the phospholipids can move around, right? The membrane is actually sort of a fluid structure, not a real solid structure. The second term here, mosaic, here's a mosaic from art, right? Mosaic tile, where you put a whole bunch of little pieces together, and then when you back up and look at it, it makes a cool picture. Mosaic means combination of parts, and so the membrane being lipid and carbohydrate and protein is made up of lots of different parts, and so it is a mosaic that is fluid in nature, meaning hence the fluid mosaic model. Last structure, but not least, that we have to mention is the cell wall. Some organisms have an outer structure outside of the cell membrane called the cell wall. We're going to see this in plants, algae, fungi, some bacteria, but again, just like chloroplasts, we never see cell walls in animals. This is a thick structure, a rigid structure, again, outside of the membrane, that is there for just further support and protection. These are mostly carbohydrate with some protein mixed in, and they are just a nice solid structure that gives the cell a really nice solid form, which is good for plants and the other organisms that have them. This is sort of like an additional wall outside of your city, or just an extra layer of protection on the outside of that cell. Plant cell walls in particular are made of a carbohydrate called cellulose that I think we mentioned back in chapter 2. This is really what makes plants harder than a lot of animals, which are kind of a soft tissue. Cellulose also is really hard to break down and digest. So any sort of paper or wood product that you see is mainly just leftover cellulose from dead plant cells. So paper and other paper products, wooden tables, wooden chairs, the wood that you use there, that's mainly cellulose. Cellulose also can't be digested by the human body, so anytime that you eat a vegetable, lettuce, beans, corn, whatever it might be, part of that doesn't get digested, and you poop that right back out, which is why you maybe have seen those kind of plant things come back out of you before, like corn kernels or whatever, and then that's because the cellulose doesn't get broken down by your body. Okay, so on that wonderful note, that is the end of our video. So plenty of structures, all of which I think you've probably heard before, and we'll do an activity here next class where we start to use these and talk about these a lot more, because they are the focus of what we're doing here over the next couple of weeks. So thanks for listening, and I'll see you guys in class.